All right, our next speaker is Jeremy Warner, who's an associate professor of hematology and oncology and biomedical informatics at Vanderbilt, and medical director of the Vanderbilt Cancer Registry, and very active in standards in this area. And Jeremy is going to talk to us about genomic standards and knowledge bases for decision support. Jeremy.
Thank you, Dr. Langlotz. Thank you to the organizers of this workshop. I also want to give a special thanks to Mia Levy, who's a member of the forum and also my colleague at Vanderbilt. So Mia actually invited me down here in 2012 when I was still a fellow in Boston to come to the Cancer Informatics workshop. And, I didn't know what to expect. It was my first time coming to the National Cancer Policy Forum. And, that workshop really solidified my realization that this was my field. I love cancer informatics, and it's been really great. So I'm going to talk today about really some preliminary steps before we get to artificial intelligence and machine learning in the area of genomics. So just a few disclosures here. So since patient access was in the title of this workshop, I did want to just have this sort of patient perspective slide. So what are three questions that every cancer patient asks? And this is anecdotal, of course, but based on my experience in the clinic. So I would say for the past 5,000 years, every cancer patient has asked, who's like me? How long have I got? And what are my options? This is not an arbitrary start because the first recorded cancer prognosis is around the year 3000 BC in the Edwin Smith Papyrus, and I'll talk about May 2001 in a second. So let's rephrase these questions into our traditional diagnosis, prognosis, and prediction. What happened in May 2001? This happened. So this was the approval of imatinib, Gleevec. So this is the first genomically directed cancer treatment and really ushered in the field of clinical genomics. From there, we've done a lot. So we've, expanded our understanding of how genomics affects prognosis. And of course, we've had a incredible increase in what I call genetically influenced cancer treatments, whether that be as a function of protein expression or starting in May 2001 as a function of actual gene mutation. So from May 2001 forward, what are the three questions that every genomically informed cancer patient asks? It's the same questions. The problem is now we have trouble saying who's like you. We have trouble telling you what your prognosis is. And sometimes the treatment options feel kind of like this. So how did we get here in the span of a short 20 years? So this was already introduced yesterday, but I'll just put it on a slide for emphasis. So precision oncology is the right patient, the right treatment, and at the right time. And in 2018, really what we're talking about is to achieve these goals is through something that we'll call molecular profiling and the use of targeted genomic agents. And I put immunotherapy in parentheses there because we're all trying to find real good biomarkers for who will and won't respond to immunotherapy, and that's evolving quickly. It's also worth thinking about, how does the process of coming to a decision in precision oncology work, and who are the players? And so this is simplified, but a similar slide was shown yesterday saying, well, there are clinician tasks and there are laboratory tasks, and some of these are shared. So as Dr. Zutter talked about yesterday, test ordering is a shared process now in many cases. The lab measures. The lab makes observations. And then really importantly is the interpretation bit, which is also shared to some degree between the pathologist and the clinician. And I'll be talking a lot more about that. There's a decision-making process where an actual treatment is chosen. And then finally, prescription and action. And the idea here, right, is that this is an iterative process. The idea is that patients who are in this sort of pipeline will perhaps not respond to a single treatment forever, but it will be some sort of repetitive process to either find new treatments before disease progresses or to determine that the treatments are working. So in the first roughly 10 years of the genomic era, we were basically learning more about genes sort of one by one and sort of an ad hoc basis. And likewise, the testing was one gene at a time. And so here's just three examples and sort of seminal papers that are relating mutations in these genes to the cancer of interest, EGFR, and the drugs gefitinib and erlotinib. KRAS is actually an inverse predictor of response, and then BRAF and the drug vemurafenib. In 2011 at Vanderbilt and then other places, there was this rollout of these gene panels, and these were between six and 10 genes, not whole exome sequencing, but definitely larger than single SNP detection. And then really in 2014, which is just 4 years ago, and onwards, we began to see these big panels. And this is one. This is called Foundation 1. This is not the whole list of genes that are tested in that panel. That's not the whole list. It's actually about 300 genes, and this might even be one of the smaller panels being offered in this realm currently. So what have we been able to do with this information? Well, one thing is we've been able to really accelerate the clinical trial domain through a mechanism called molecular basket trials. And this is a great review article of those, but basically the idea is that now you are assigning patients to interventions based on some combination of a traditional cancer diagnosis as well as their genetic makeup. Probably the most well-known of these is the NCI MATCH trial, which has been accruing very quickly and successfully. Another one I wanted to mention is the ASCO TAPER trial, which stands for Targeted Agent and Profiling Utilization Registry Study. This trial, some of the nice things about these kind of trials is that they really engage the community. So in order to find enough patients to enroll in the various arms of these basket trials, it's not sufficient to be open at 1 or even 30 academic centers. So TAPER, for example, is open at 113 sites across 20 states. And just as importantly, these types of trials, in order to be successful, really have to collaborate with multiple pharmaceutical industry partners. And as you can see, ASCO has 7 pharma partners in order to bring the diversity of drugs to the patients. So over at the FDA, obviously, folks are aware of the evolution of precision oncology, but I do want to point out that 2017 was a banner year in the fact that of the approvals that were gene mutation specific, which is to say that the label actually has a particular mutation, gene and mutation requirement, all of them in the past year were tied to the label as a requirement for the prescription of that drug. So really a watershed moment. And then there were these 2, what I call game changers that happened last year. So one of them happened in May, and this was the first site agnostic approval of a cancer therapy. And this therapy is immunotherapy called pembrolizumab, and the FDA approved this for any solid tumor with a certain genetic finding. The second game changer that happened last year was in November, and this was also from the FDA, the first approved companion diagnostic gene panel. And as many alluded to yesterday, many of the innovations in oncology do not take place until the alignment of the payment incentives. And I believe that this is probably an even more important game changer in that we will see a vastly increased uptake of these large gene panels in the coming year. So where are we at right now in the clinic with what we get back as clinicians when we order these panels? Well, it's far from standardized. And here are just 3 examples of reports that come back. We don't need to dwell on the specifics except to notice that this one is grainy and black and white because it's a fax. So the very sophisticated molecular profiling company that generated this and spent thousands of dollars and many, many curators ultimately it got scanned in and faxed to the clinician. And then here's another. And you can see that there's really no standardization in the way these reports appear. And then to drive home that issue, we asked a couple years ago through a survey of ASCO members, when you order one of these gene panels, does the lab you're ordering from send, and can your EHR receive that data as a discrete data element? And less than a quarter of the respondents said so. So obviously, if we're going to use artificial intelligence in this space, we need to move beyond this. And so at Vanderbilt, we did work with with a partner lab to move from this scanned black and white image to at least their full color PDF image, and also an electronic representation of some of the data in that report, so that we could not only insert it into our EHR, but also use it for potentially clinical decision support. We also, so I should, emphasize that process was what you call one-off, so it was not generalizable. Subsequently, we actually started working through Health Level 7 International to start to define standards so that we could move clinical genomic information between sites and enable things like third-party apps. And this was our first iteration called Smart on Fire Genomics, and it has evolved. We don't need to dwell on the details, but basically we're breaking out each of the potentially clinically important genetic findings and describing how it should be represented. This has also evolved in the most recent sort of encapsulation of this genomic standard is in what's called an HL7 domain analysis model. So if we can standardize genomics, we can actually have a workflow like this, which is potentially completely digital. So you've got up at the top, you've got your clinician ordering a genetic test. At the bottom, you've got a sequencing lab performing the test and returning the results back to the clinician. And then off to the side there, you've got the potential for third-party applications that can present and contextualize genetic test results. This is just a report from the President Cancer Panel that came out that emphasized that precision oncology applications, such as the one I just showed you, could be a solution to what they said was a real need, which was tools such as apps that could help the oncology workforce. So beginning last year, the ONC sponsored a program called Sync for Genes, which sought to accelerate this standardization even quicker. And basically, there were 5 pilot sites that address different aspects of genomics in clinical care. We collaborated with Foundation Medicine on this area of somatic molecular tumor next-generation sequencing. And then others participated in other aspects of getting clinical genomics into a standard form. So transitioning just a bit here to, so if we achieve the standardization of genomic data, we still need these knowledge bases, and why? So the fact that I can show you this graph is only because I could receive we received that data digitally, but you can see quite a issue here. Of the results that we're getting, and this is real clinical results, only a third or less than a third of those are what have been labeled as actionable by the generating lab, and the other 2/3 are of unknown significance. A secondary problem is that thinking of these mutations in isolation is not correct. The median number of what actionable variants that we're getting per patient is actually around 4, and the median number of unknown variants is about 7. And so this is a challenge. And then a related challenge, and AACR Genie was mentioned before. So this is data from the first public release of Genie, which is about 17,000 patients tested in the clinic with genomic testing. And this is just a histogram of the top 100 most frequent genetic variants in Genie. So 2 things I'll ask you to notice. One is that the y-axis tops out at less than 1%. So the most common mutation in this database is about 0.5% of the patients. Incidentally, that mutation is not actionable, or it's sort of inversely actionable, in that it indicates that you should not give a treatment. And then you see this fall off, and this is not a typo, that little pixel there. In fact, the top 100 variants only account for 10% of the distinct variants in Genie, and then there's another about 79,000. These are unique variants in that database. So to think that a clinician or anybody, any human could digest even a fraction of that information, I think is impossible. So we need knowledge bases, and we need artificial intelligence. So, talking about the knowledge bases that are out there currently, so these are probably the most well-known. There are others, but these are for the most part human curated. So some of them may bring in some knowledge engineering from computational sciences, but for the most part, these are based on human curation. But they're uneven, so we've done some comparison of the content of these knowledge bases, and we compared two cancer-specific knowledge bases, OncoKB and Civic, and then a third broader genomic knowledge base called ClinVar, and there's some overlap at the gene level, but there's a lot of uniqueness to each of these knowledge bases. And even when you look at an enriched gene list for cancer, the cancer-specific ones still have their own gene, and this is gene level, not even variant level, genes that only they have in that knowledge base. Additionally, one of the great things that these knowledge bases are doing is structuring this data. So it is human curated, but it's being fit into models that are potentially computable. The problem is that each of these knowledge bases has a slightly different model. For example, the definition of this one, Civic, says disease, this one says tumor, and they don't quite match up the concepts. So mapping across these knowledge bases is no small task. I did want to call out the Variant Interpretation for Cancer Consortium, which is working on this task. So the idea being that everybody should agree to a common model, or at least a minimal common set of elements that should be more or less equivalent across these knowledge bases. So, taking all that in, last fall we published a set of recommendations that are really intended to facilitate the adoption of genomic medicine. And this was based on a workshop that ASCO held about a year before. And I'll just emphasize, there's a lot of text here, I'll just emphasize the bold. So we said genomic, oops, how do I go back? Can you take me back one slide? Yeah. So we said, we should use the past tense. I mean, genomics knowledge has already grown beyond human capabilities, and I hope you agree with that after seeing some of that information I just showed you. But we need solutions. We need comprehensive standards, and most importantly, we need standards that are harmonized and accepted by all, and all means basically the data producers and the data consumers. And then furthermore, I didn't get to talk much about APIs, but any genomic knowledge base that's making its content freely available, and they don't all do, but many of them do, should at least consider how to make that knowledge available through APIs so that it can be consumed by systems such as EHRs or to enable clinical decision support. So my take-home points are that genomics will become increasingly relevant. I think we're at an inflection point now with that approval to cancer care, not only during and after diagnosis, but potentially before. If you've seen the data on that CancerSeq test about potentially using genomics to diagnose cancer as well. But without agreed upon standards, we're not going to be able to build large data sets that inform diagnosis, prognosis, and treatment. And likewise, given the vast breadth of knowledge that we're dealing with here, we need artificial intelligence and machine learning to be able to practice precision oncology. With that, I'd like to acknowledge all these folks, and thank you.